and off. And at this point, we're going to go into our last few presentations here. So please welcome the team from Louisa County High School.
Good afternoon. My name is Aaliyah.
I'm Michael.
I'm Kelly,
And I'm Catherine.
and we are presenting the effects of magnesium sulfate on experimental Arabidopsis thaliana plants. Arabidopsis thaliana is a good model organism for genetic research due to its small genome and quick reproductive cycle, which allows for easy genetic variation. In some of the Arabidopsis thaliana plants, the AADL, or allyl alcohol dehydrogenase-like gene, was disabled. This gene behaves much like a flavonoid, particularly anthocyanin, which is responsible for pigmentation change in plants.
Since the AADL gene was disabled using insertional mutagenesis, there was little to no restriction of how much UV light was able to penetrate the plants. This affected chlorophyll because chlorophyll contains the magnesium ion, which increases the rate of photosynthesis. Magnesium deficiency can be seen by a yellow discoloration in the leaves and veins of A. thaliana. We decided that a supplement of magnesium sulfate would help decrease the chance of the magnesium deficiency and aid in root growth of experimental A. thaliana.
It was hypothesized that if soluble magnesium sulfate was added to the soil during phase 1, then the mutant experimental plants would appear a darker hue than the wild-type experimental plants.
At the beginning of the experiment, 1,400 milliliters of water was added to unfertilized soil. Magnesium sulfate was added immediately after the cotyledons emerged, and all experimental plants were watered with a 2 molar concentration of magnesium sulfate. Rosette length and plant height were measured 3 times a week. The plants were under a 12-hour photo period, but this was eventually changed to 24 hours as an attempt to force bolting. So this graph shows the plant stem heights of all 4 Arabidopsis thaliana groups. As you can see, the mutant experimental plants have the highest plant stem averages of all these groups. This is significant because even though all of the experimentals did die, the mutants still managed to have a pretty high stem height growth. Similarly, all controls and all experimentals did not show to have a steady growth pattern, and all experimentals averages decreased drastically due to plant mortality.
The stem heights of the mutant experimental and mutant control plants fluctuated throughout the experiment, with the more sporadic experimental plants having a margin of 1.5 millimeters from the initial height, and the more consistent and steady control plants having a 1 millimeter margin from the initial height.
This graph shows the stem height of wild-type and mutant experimental A. thaliana. It was seen that both plants experienced sporadic growth patterns and increase and decrease of height. Both plants began to drastically decrease toward the end of the experiment because of an excessive buildup of magnesium sulfate on the soil, which caused a drought in the plants, which in turn caused oxidation.
This graph shows the overall rosette height of all of the Arabidopsis thaliana, all of the rosette length of all of the Arabidopsis thaliana plants and their growth throughout the experiment. As you can see, both types of control plants exhibited steady and comparable rosette growth, while the experimentals, on the other hand, experienced a lot of fluctuating growth. This fluctuation is due to the decreased trigger pressure and curling of the rosette leaves, which eventually led to a decrease in average rosette length.
The rosette length of the mutant control and mutant experimental plants was relatively similar and stayed relatively equal throughout the first 17 days. However, after day 17, the mutant experimental plants experienced a large decrease in growth rate. On the other hand, the mutant controls experienced a large increase in growth rate than the more sporadic experimentals.
This graph shows the rosette length of wild-type and mutant experimental A. thaliana. While both plants experienced sporadic growth patterns, the wild-type experimental showed a more steadier rate of growth and larger rosette leaves measured,
until both plants begin to rapidly decrease 34 days after planting. The image on the left is of a mutant control pot 7 weeks after planting. It shows that all rosette leaves have turned a dark purple pigmentation. The image on the right is of a wild type control pot 7 weeks after planting. It shows a small rosette leaf that was uncovered from underneath a larger rosette leaf to still have a bright green pigmentation. In group 1, this was observed on plants 3, 4, and 6, and also in other groups' plants. This was due to the incorrect photoperiod that was applied at the beginning of the experiment. The images on the left and right show holes and cuts in the rosette leaves. This was caused by small organisms that were in the soil, such as protozoa and actinomyces, which are responsible for the breakdown of organic matter such as sulfur.
These images show a significant amount of magnesium sulfate buildup on both types of experimental plants. The reason for this was because of the high concentration of magnesium sulfate that was added to the solution that was given to the plants. The magnesium sulfate did not evaporate from the supersaturated soil, which caused a significant crusting and therefore led to a decrease in the sugar pressure of the experimental plant leaves. Throughout the experiment, all of the Arabidopsis thaliana plants experienced a change in pigmentation, but it was especially evident in the control plants. As you can see, the wild-type control plants exhibited a purple coloration in the stems 2 to 4 days before the mutant-type control plants exhibited this coloration. Although both plants eventually changed to the same pigmentation towards the end of the experiment, the wild-type control plants did so quicker, and it was more evident quicker.
According to NASA, visible light ranges from about 400 nanometers to 700 nanometers. However, plants also absorb light energy from above that from light energy like infrared, which is above 700 nanometers. Through the use of a spectrophotometer, we were able to determine that when subjected to 600 nanometers of light energy, the mutant experimentals would absorb more, 14% of the energy, than the wild-type experimentals, which only absorb 9.1% of the energy. However, when subjected to 800 nanometers, the mutant experimental absorbed 10.3% of the energy, which was 6% less than the wild-type experiment.
The hypothesis of the experiment was rejected initially because the results of the experiment concluded that the mutant experimental plants were not a darker saturation than the wild-type experimental plants. The wild-type experimental plants were actually a darker saturation because they became a darker green and eventually turned white in color and died, while the mutant experimentals remained a lighter green before also turning white and dying as well. But this significantly happened after the wild-type had made their transition. This delayed transition was due to the AADL gene's inability to allow the wild-type plants to adapt to the large quantities of magnesium sulfate in the soil, thus producing a higher plant mortality rate.
However, later it was determined that the quantitative data, rather than the qualitative data, that was provided by the use of the spectrophotometer supported our hypothesis. This was because when the mutant experimental absorbed 14% of the energy, while the wild-type experimental only absorbed 9.1% of the energy. This was when it was subjected to 600 nanometers, which is within the visible light spectrum.
We would like to thank Dr. Lally and Virginia Tech for partnering with us to conduct our Arabidopsis thaliana experiment. Now the field is open for any questions. The magnesium sulfate was mixed in a 2 molar concentration, and it was applied to the top of the plants twice a year.
The leaves?
Well, it was applied, we applied it to the soil, and we're very careful not to touch the leaves.
So like in your graphs, I saw that like at some points plants would like get smaller and then go back up. What was going on?
So what was happening at those points was their turgor pressure, which is the amount of pressure that the water can cause, was less than it was earlier, which was causing it to appear to have like shrunk essentially, and that was caused by the evaporation of the water due to the increased evaporation of water due to the magnesium sulfate.
Also, turgor pressure is another word for wilting. That wasn't clear. Loss of turgor pressure, another word.
How often did you live with
Minus the magnesium sulfate. With it, okay, so our plants were watered on Tuesday and Friday of every week with 25 milliliters of water with a 2 molar concentration of magnesium sulfate.
So if like you don't know what a molar concentration is, in each 25 milliliters of water, there was 1.5 grams of magnesium sulfate.